I am Dr. W. Gunode from Kamla Nehru Mahavidyalaya, Nagpur. Today we are going to discuss the nucleic acid, and as we know, nucleic acid is the important constituent of the biopolymers, and it is a constitute three parts. One is a pentose sugar, then a nitrogenous base, and the phosphoric acid. So these are the main three constituents of the nucleic acid, and here we are going to discuss what are the different constituents of the nucleic acid. After that, the structure of primary structure and secondary structure of nucleic acid, and also what is meant by the nucleotide and nucleosides. All these aspects we are going to discuss in today's presentation. So the today's topic is the nucleic acid, and in nucleic acid we are discussing the first of all what is the nucleic acid and what are the constituents of nucleic acid. And in this case, as we know that nucleic acid constitute important class of biomolecules which are found in the nuclei of all living cells in the form of nucleoprotein, that is the protein which contain nucleic acid as the prostatic group. The name of nucleic acid was given to this class of macromolecule since first nucleic acid was isolated from nucleus of the first cell and was found to be acidic in nature. Nucleic acid are the genetic material of the cell and are responsible for transmission of hereditary effect from one generation to other. They also carry out biosynthesis of protein, and the genetic information coded in nucleic acid control the structure of all protein including enzyme, and also govern the entire metabolic activity in the living organisms. Therefore, we can say that nucleic acid are biopolymers, that is the polymer present in the living system. They are also called polynucleotides since the repeating structural unit, that is monomer unit of the nucleic acid, is a nucleotide. Each nucleotide is made up of three parts, that is a sugar molecule, a heterocyclic nitrogenous base, and the phosphoric acid. So these are the three parts of this nucleic acid, and here is shown three parts of this nucleotide is made up of three parts: a sugar molecule, heterocyclic nitrogenous base, and phosphoric acid. And so in general we can say that nucleotide may be represented as a in this way. Nucleotide can consist of three parts, and here this is the phosphoric acid, this is sugar and base. So they are linked together in this way, and therefore nucleic acid may be represented in this way. It is a biopolymer, and so here is the long chain. This is one unit, that is the base, then sugar, and then phosphoric acid. Again this combined with the sugar, base, and phosphoric acid. In this way, large number of molecules are monomeric unit joined together and forming the biopolymer which is known as nucleic acid. So here is shown the example of nucleotide, how it is represented, and how the nucleic acid form the polymers. And now what are the constituents of nucleic acid? So in this case we can say that all nucleic acids on complete hydrolysis give the mixture of three different types of compound. And these three compounds are: one is a phosphoric acid, second is a sugar, and third is a number of nitrogenous bases. And phosphoric acid SCPO3, and second is the sugar. So sugar, in this case two sugars have been isolated from hydrolysis of nucleic acid. Both are pentose, that is D-ribose and 2-deoxyribose. Both these sugars are found to be a furanous form as shown. This is the furanous form of the D-ribose and 2-deoxyribose. So here is shown furanous forms of D-ribose and 2-deoxyribose. So this is the structure of the D-ribose, and this is the 2-deoxy-D-ribose. So this is furanous form of the sugar, and third is the number of nitrogenous bases. So number of bases are here. And as we know that in case of bases, there are two different classes of heterocyclic nitrogenous bases which have been isolated by the hydrolysis of nucleic acid. And these two bases are, one is a purine and second is a pyrimidine. And in this case purine, first is the purine, and in this purine, two purine which are most commonly found shown here, and these are found in the nucleic acid. These two are adenine and guanine. So structure of this adenine and guanine is shown here. So this is the structure of adenine, and this is the guanine. So and this is the heterocyclic. Nitrogen is here present in the ring. So this is the heterocyclic here, and only the bonds are changed, and the similar structure is here. This is the regional structures of these two. This is also show guanine, the regional structure, and so this adenine and guanine having this type of structure, and these are the purines. And second is a pyrimidine. So pyrimidine have this type of structure, and here most commonly in this case the pyrimidines in nucleic acid found are uracil, thymine and cytosine, and the structure of uracil occur generally only in the RNA. This is the uracil, short is a U, occur only in the RNA. This is thymine, occur in the DNA, and this cytosine occur both in DNA and RNA. So these are the structures of pyrimidine, and three pyrimidines are there. One is a uracil, thymine and cytosine. So these are the constituents of the nucleic acid, and three parts are there: phosphoric acid, sugar and the bases. And here structure of purine also here shown, already shown, and now here nucleoside and nucleotide are the two important part in the nucleic acid. So what is meant by the nucleoside and what is meant by nucleotide, that we are going to see here now. And in case of nucleoside, this nucleoside contain only two basic component, and these two base of the nucleic acid, and these are pentose sugar and the nitrogenous base. These are the two. So nucleoside contain two parts. One is a pentose sugar and the nitrogenous base. In general, we can say that nucleoside may be represented as sugar base, like sugar base. So during their formation, the carbon atom in this case sugar are numbered as carbon atom in the sugar are numbered as 1, 2, 3, 4, 5 dash etc. in order to distinguish them from the numbering of the atoms of the bases. Depending upon the type of sugar present, nucleosides are two types. So these nucleosides are two types. One is ribonucleosides and second is deoxyribonucleoside. And the name of ribonucleosides are 5 bases. So ribonucleosides of the 5 bases are shown in the table. These bases are, one is adenine, guanine, cytosine, thymine, uracil. Their abbreviation A, G, C, T, U. And nucleosides are adenosine, guanosine, cytosine and thymidine and uridine. So these are the bases shown here, and the name of the deoxyribonucleoside of the 5 bases are obtained by adding the prefix deoxy to the name of corresponding nucleoside, ribonucleoside, and these are deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine and deoxyuridine. So prefix you have to add in the beginning, and that is the deoxy. So there are also these 5 deoxyribonucleoside can be represented. For example, here is shown structure, that is the name of the shown here in this way. For example, adenosine is a nucleoside formed by the combination of cytosine, pyrimidine base and 2-deoxyderibose sugar as shown below. So this is obtained by adenosine is obtained by the combination of two here, adenine and the ribose. So this is adenosine, the combination of these two, and here is a cytosine and deoxyribose and forming deoxycytidine. So these are the bases formed. So here we can say that the deoxycytidine is a nucleoside formed by the combination of cytosine and the 2-deoxyderibose as shown here. So this is the first, that is the nucleoside, and consists of two parts, and the second part is the nucleotide, so which is shown here. Both are the important, and in case of nucleotide we can say that a nucleotide contains all the 3 basic components. So this is important, this is the main part, and nucleotide consists of it contains all the 3 basic components of the nucleic acid, and these basic components are phosphoric acid, pentose sugar and nitrogenous base. These are formed by esterification of one of the hydroxyl group of the nucleoside with the phosphoric acid. In other words, nucleotide are nucleoside monophosphate. So it is known as nucleotides are nucleoside monophosphate. So in this way, and depending upon the type of sugar present, nucleotides like nucleosides are also 2 types. So nucleotide are also two types. One is a ribonucleotide, second is deoxyribonucleoside types. And here nucleotides are abbreviated by 3 capital letter, and here it is shown, for example, these are shown short forms: AMP, adenosine monophosphate. Second is DMP, deoxyadenosine monophosphate. UDP, uridine diphosphate. ATP, adenosine triphosphate. So these are the abbreviation in capital letter, 3 letters. And here structure is also shown of AMP and also UDP. So these are the structures shown here. This is the structure of adenosine monophosphate, and this is uridine diphosphate. This is AMP, this is UDP. So structure of these two are shown here, and of the nucleotide. And also, as we know that di, tri, oligo and polynucleotide are obtained by joining together 2, 3 up to 10 and also thousands of nucleotide through the phosphodiester linkage between 5 and 3rd carbon atoms of the pentose sugar. So they can be joined in this way here, and to the carbon number 5 and also carbon number 3, they joined and forming this type of nucleotides. So yeah, it is possible to join 2, 3, 4 or many molecules can be joined together to the through the phosphodiester linkage. And so these are the two important terms. One is a nucleoside and second is a nucleotides. These two terms are very important, and their types, 2 types are there. After that we are going to see what are the types of nucleic acid. So as we know, nucleic acids, depending upon the type of sugar present, nucleic acid are 2 types. One is a deoxyribonucleic acid and second is a ribonucleic acid. In short, it is known as DNA and RNA. So DNA, what is meant by DNA? So the difference between these two are given, shown here some points. First point shows that the sugar present in DNA is the 2-deoxy-D-riboz. In case of ribonucleic RNA, sugar present is a RNA, that is D-riboz. And second point is DNA contain cytosine and thymine, that we have seen, as a pyrimidine basis, and guanine, adenine as a purine basis. In case of RNA, it contains cytosine and uracil as pyrimidine basis, and guanine and adenine as a purine basis. In case of DNA, has double standard alpha helix structure. RNA has single standard alpha helix structure. So there is a difference between DNA and RNA. DNA chiefly occurs in the nucleus of the cell. RNA mainly occur in the cytoplasm of the cell. DNA molecules are very large. Their molecules weights may vary from 6 million to 16 million. RNA molecules are much smaller, with molecular weight ranging from 20,000 to 40,000. So it is smaller, and DNA is larger. DNA has unique property of replication. RNA does not have replicate. DNA control the transmission of hereditary effects. RNA control the synthesis of protein. So there is a difference between deoxyribonucleic acid and ribonucleic acid. So these are some points of differences between these two types of nucleic acid. So there are 2 types. One is DNA and second is RNA. That is the 2 types of nucleic acid. And also there are primary structure and secondary structure of the nucleic acid. And in case of primary structure, we can say that 2 structures are there. And first is the primary structure, and nucleic acid are polynucleotide. They are formed by the combination of thousands of molecules of nucleotide with the elimination of water molecule. During this polymerization, the hydroxyl group of the sugar residue of one nucleotide combined with the hydroxyl group of the phosphoric acid group present on the carbon 3 of the other nucleotide to form long polynucleotide chain. In this way, during this combination, the backbone of nucleic acid consists of alternating sugar phosphate residue. Each sugar on this backbone is further connected to one of the 4 nitrogenous bases, and therefore the sequence in which 4 nitrogen bases are attached to the sugar phosphate backbone of the nucleotide chain is known as the primary structure of nucleic acid. I am repeating again that the sequence in which the 4 nitrogen bases are attached to the sugar phosphate backbone of the nucleotide chain, this is known as primary structure of this nucleic acid. And structure is also shown here in this way. This is the primary structure, and in this structure is shown, and from the here it is also evident that time consuming to write the complete primary structure of nucleic acid chain and even oligonucleotide. In order to simplify, the bases are represented by their one letter symbol, that is A, C, G, T, U. The phosphate bond is represented by the symbol P, and the sugar is drawn by the simple Fischer projection. So this is the structure as shown, and as we know that nucleotide, tetranucleotide having adenine, cytosine, guanine, thymine bases end is represented as ACGT as shown in this figure. So these are the symbols ACGT, and how here third carbon, fifth carbon, P is the phosphoric acid, and these are the chain formed in this way. So they join one by one and forming the long chain, and this is the primary structure of this nucleic acid. And there is also secondary structure, and in case of secondary structure we can say that the secondary structure in case of secondary structure of nucleic acid, the DNA consists of two strands of polynucleotide and coiled around each other in the form of double helix. The backbone of each strand consists of sugar phosphate unit, and the base unit of each strand are pointed into the interior of the helix and are linked together through the hydrogen bond, whereas G and C are held by 3 hydrogen atoms. A and T are held by 2 hydrogen atoms. Unlike DNA, RNA has a single strand. So this is the structure is shown, different structure of this compound, and here first structure is the double helix structure of DNA shown here. This is the helical structure, double helix structure shown here for this DNA, and the 2 nucleotide strands are shown here, and the 2 nucleotide strands are held together by specific hydrogen bonding between the bases. The numerical, the 5 star and 3 star indicate the free hydroxyl group of the terminal deoxyribose unit and present at the 5th carbon and 3rd carbon position is shown here. So in this way, here also distance is shown. So this is the distance between these two. Here this is the coil type structure. See here in nanometer structure is shown. This is the 5th carbon, this is 3rd carbon. So they are coiled, this helix like structure is obtained, and these are the pyridine bases are there, pyrimidine bases are here. So they join in this way and forming this type of structure. Another structure, pairing of complementary bases shown here in the second figure. It is shown that the structure, 2 strands are joined together. Here see, now here this is the sugar, in this case sugar, this is adenine, hydrogen bondings, these 2 molecules are joined by the hydrogen bonds. This is hydrogen bond, again sugar is here. In this way, different structure A and B. A is a thymine with the adenine through the hydrogen bonds, and cytosine. B is the structure which is shown the cytosine and guanine through the hydrogen bond. So in this way 2 structures have been shown, and these structures are known as the secondary structure of the nucleic acid. So primary structure and secondary structure, what is DNA, what is RNA, difference is also we have seen. So today we have discussed the detail about the nucleic acid, and what is nucleic, as we have seen what is nucleic acid. It is the biopolymers, and it constitutes 3 important parts. One is the heterogeneous, that is heterocyclic base, that is the, and also the phosphoric acid, and third is the sugar. So all these 3 are the important part, and we have also seen what are the different that is the primary structure and secondary structure of nucleic acid, and also we have seen the important 2 part, that is nucleotide, nucleosides. So nucleoside, it consists of 2 part, and the nucleotide consists of important 3 part, that is the base, sugar, and the phosphoric acid. So these 3 important parts, and so there are 2 types of nucleic acid. One is a DNA, other is RNA. That difference between these 2 also we have discussed in this nucleic acid presentation.